Hello, welcome to another edition of 5-Minute FRQ, where I take you through an old FRQ on the AP Bio exam in 5 minutes or less. As always, this information is freely available on the College Board's AP Bio website, so if you have any questions, you can look it up. This is question 6 on the 2019 AP exam, and the question begins with a data table here that might not make any sense to you at all, so let's just skip it and read the prompt and see what's in there first. All right, the yeast Saccharomyces cerevisiae is a single-celled organism. Amino acid synthesis in yeast cells occurs through metabolic pathways, and enzymes in the synthesis pathways are encoded by different genes. The synthesis of a particular amino acid can be prevented by mutation of a gene encoding an enzyme in the required pathway. A researcher conducted an experiment to determine the ability of yeast to grow on media that differ in amino acid content. Yeast can grow as both haploid and diploid cells. The researcher tested 2 different haploid yeast strains, mutant 1 and mutant 2, each of which has a single recessive mutation, and a haploid wild-type strain. The resulting data are shown in Table 1. Okay, there's a lot going on here. If you're familiar with the Beadle and Tatum experiments, where they were using, I think, bread mold, and they were knocking out different genes. This is sort of a variation on that. So if you know that experiment, that's helpful, but you didn't have to know it to get this question right. So under the table, the caption says, the data show the growth of haploid Saccharomyces cerevisiae yeast strains on media that differ in amino acid content. A plus sign indicates that the yeast strains grow, and a minus sign indicates that the strains do not grow. Okay, so I have 4 different treatments. Treatment 1, I give all amino acids. Treatment 2, I give none. Treatment 3, I'm missing methionine. Treatment 4, I'm missing leucine. And I have 3 strains, the wild type, the mutant 1, and the mutant 2. Okay, the way you interpret this chart, what this is basically trying to tell you is which genes have been knocked out. So my wild type grow in everything, all right? Mutant 1, when I give it all the amino acids, it grows. If you look at treatment 3, when I don't give it methionine, it doesn't grow. And that tells you that treatment 3 tells you that mutant 1 cannot make methionine, all right? Treatment 4, mutant 2 can't grow. With treatment 4, I don't give it leucine. And treatment 4 is telling you that mutant 2 cannot make leucine, all right? Treatment 2 with no amino acids, neither mutants grow because they can't make either methionine or leucine depending on which treatment. Okay, this is a difficult question. So question A, identify the role of treatment 1. Treatment 1 is the control. All right, that's all you had to say. Looking at the rubric, if you simply said it's the control, then that's perfect. The deal with a positive control versus a negative control, a positive control is a treatment where you're making sure everything works. So my variable here is what amino acids do I have? Well, I gave the strains all of them to make sure that they grow. If I gave all of them and they didn't grow, something else is going on. So a positive control basically tests to make sure your setup is working. But if you just said control, that worked. B, provide reasoning to explain how mutant 1 can grow on treatment 1, but cannot grow on treatment 3, because it can't make methionine. All right, if you look at the rubric, it simply says, mutant 1 can use methionine when it's present, but mutant 1 cannot synthesize methionine because you knocked it out. All right? That's where knowing the Beadle and Tatum experiment is helpful. Part C, yeast mate by fusing 2 haploid cells to make a diploid cell. In a second experiment, the researcher mates the mutant 1 and mutant 2 haploid strains to produce diploid cells. Using the table provided, predict whether the diploid cells will grow on each of the 4 media. Use a plus sign or minus sign to indicate growth, and use a plus sign to indicate growth and a minus sign to indicate no growth. So in this chart here, you have to add pluses or minuses. All of them should be pluses, all right? Treatment 3 shows that mutant 1 can't make methionine. Treatment 4 shows that 2 can't make leucine. Those are haploid. If I put them together, and the new strain has 2 copies of each, then what one couldn't make, the other one's providing the gene for. So this last column would have growth in all of them because the genes I knocked out were either methionine making in treatment or in mutant 1 or leucine in treatment in mutant 2. If I hybridize them, they can make everything. So they'll grow in every column. Okay, hope that was helpful, and I'll see you guys next time.